Welcome everybody. Thank you so much for joining us today. My name is Varun Narayan. I am a technical account manager here at LabKey. And today we're going to be talking about our Biologics LIMS product and how it's going to help you unify and connect your R&D data for faster decisions. So we're going to spend a little bit of time talking about the problems that people are facing in the scientific community. We'll highlight some of the features of Biologics LIMS. But for the main bulk of this webinar, we're actually going to be jumping into the product itself and then showing you how it works and kind of what's possible within the product. But kind of first off here, I wanted to let you all know a little bit more about who we are at LabKey. So, we've been around for over 20 years, and we've used that experience to provide scalable solutions to our clients in a variety of different sectors. So, from academia and research, all the way to biotech and pharma. And really, when it comes to us identifying new features, new products. We heavily rely on feedback and guidance from organizations just like yours. And honestly, that's how our Biologics LIMS product kind of came about. We had heard time and time again about these real issues that people were facing in the scientific community that had kind of little to no real solutions. Um, so let's kind of touch on those real quickly. So the first of these is that complex entities and construct relationships are hard to represent in generic tools. So, we understand that your data is unique to you with all of its eccentricities. And trying to force it into kind of a rigid, predefined structure can oftentimes be more trouble than it's worth. Uh, and honestly, you may find a solution that works for some of your data, but not all of it. And so you end up kind of Frankensteining multiple kind of solutions together in order to get that big picture. And that can actually lead directly to the next problem, which is that having your data scattered across these multiple spreadsheets and different systems actually blocks asking these cross-project questions. So if you manage to finally find the right data that you need, being able to share it with other teams, other groups is pretty complicated unless you want to spend kind of a lot of time manually piecing things together. And having to manually piece these things together and perform these manual handoffs increases errors and it slows down your ability to make these quick decisions. So each extra step introduces yet another opportunity for human error and slows things down even further. And especially kind of when it comes time to be able to quickly identify what's working, what's not, what's viable, what to pursue. These slowdowns can be incredibly detrimental to your process. And finally, traceability gaps create risk during investigations, tech transfers, and documentation. If you can't ask questions of your own data and see who did what, when, and why, you're not going to be able to get the repeatable results that you're after. And you're not going to be able to train team members on how to perform the actual kind of day-to-day activities that you're performing in your labs. So, with all of that in mind, we created our Biologics LIMS product. So, Biologics LIMS is purpose-built to model biologics entities, constructs, and relationships. So it has the ability to model your antibodies, multi-specifics, fusion proteins, while also kind of capturing the sequences that go alongside your data as well. So no matter what kind of biological entities you're working with, being able to register all of that data in the same place is incredibly powerful. And being able to register it in whatever kind of variety of ways that your processes work, whether that's utilizing the UI or utilizing file imports or utilizing APIs programmatically. Being able to easily consolidate the data that you have for whatever kind of entities you're working with in one place is something that's kind of baked in to the Biologics LIMS product. Not only that, but it's also a unified system of record for your entities, samples, experiments, and results. So not only can you capture kind of all of the biological entities you're working with, but you also are able to kind of link that alongside your samples and your experiments to get this kind of holistic view and idea of your data, which allows you to answer those cross-study questions. It allows you to perform comparisons on your data and also kind of analyze trends over time. It allows you to standardize complex workflows while staying configurable to how your lab runs at the same time. So you can create repeatable templates that allow you to easily standardize your most kind of routine processes and cut down on avenues for human error. But it's also flexible enough to grow with your own team. So as you kind of add modalities or add new groups or sites, the software is flexible enough to adapt to these new workflows and new processes that you're going to be utilizing as well. And it also enables traceability, visibility, and insight across your programs. So it gives you that defensible who did what, when, and with which materials idea that we were talking about earlier, and allows you to have a clean, I guess, insight into exactly what's happening with your data. So, with all that being said, I want to spend kind of the bulk of the rest of our time here diving directly into the product and giving you a sense of what's possible. So, here, let me grab this page, make sure I'm sharing the right thing, drag that over. Okay, perfect. So, we're going to start off here on a molecule. So, we've got Herceptin Fab. So right now we're looking at kind of the molecular overview, molecular details page. You're not kind of limited to registering molecular data by any means. You can also capture data on a variety of other entities, such as vectors, cell lines, constructs, including the actual protein sequences and nucleotide sequences that comprise your molecules. So let's actually kind of dive right into a heavy chain here. And an aspect of the Biologics LIMS product is that kind of no matter how you are receiving the sequencing information, so that's whether you're performing the sequencing in your lab or you're contracting that out to a third party, regardless of how you are receiving these sequences, you can register them right into the product alongside any annotations that you're tracking as well. So like your frameworks, CDRs, leader regions, things like that. And you can have that sequencing information right alongside physical properties of these sequences as well. So things like average mass, isoelectric point, and things of that nature. And from here, I can kind of drill down even further. I can go down into the nucleotide sequence that kind of comprises the protein sequence. Or I can jump back up the tree, walk back up to the molecule, or I could explore any other kind of related entities here. So as you can see, you can capture kind of a variety of metadata about these biological entities. But you can also capture some information about how that data is kind of intricately linked. So you can model those relationships. And we also kind of provide an easy and kind of intuitive way to visually represent these linkages as well. So here, if I jump over to my lineage tab, here, just at a glance, I can get a view of how my data is connected. So right here, I've got my molecule, Herceptin Fab. But I can also see the nucleotide sequences and protein sequences that make up my molecule. And then downstream, I can see the expression systems that this molecule is associated with. And even further down, I can even see the protein batches, the samples that these expression systems are generating. And just by clicking around, I can actually see my metadata around these entities as well. So here's my molecule, my protein sequences, my nucleotide sequences. So you can kind of capture just at a glance, capture and understand how all of this data is related to one another. And obviously, this kind of linkages and this idea of relationships isn't just reserved for the biological entities. It also kind of extends down to the sample level as well, which you can see here. And this information is also tracked just kind of on the overview page as well. So here you can see that we've got 40 protein batches that are associated with our molecule. And so let's actually dive right into these samples. And so here we're looking at a sample type grid. And as you can see, there is a variety of metadata captured here as well. So we've got some information about where these samples are stored. So in kind of freezers that have been configured in Biologics LIMS. And I've also got some information here about the expression systems and molecules that these samples are associated with as well. And this grid is kind of fully customizable. So you can add more fields that are relevant to your use case. You can hide fields that maybe you don't want to see at any kind of given moment. So it's really easy to kind of tailor make this grid and this view to the exact type of science that you're performing in your labs. Um, and kind of model your actual processes that you're working with. And speaking of those processes, one of those might be generating these protein batches and then characterizing them. So let's kind of walk through what that process would be in our system. So here, let's go ahead and select 5 of these, and then let's add them to a plate. So we're just going to be kind of walking through a process of, creating an activity assay for these protein batches. So let's give it a pretty generic name here and today's date. And then I'm actually going to be adding these to a pre-configured template that I've created. So here we go. We're gonna hit preview, and I'm going to make this a set of 4 plates. So let's get this all configured. Go, and then I'm just going to give these some generic names for the sake of this demo. So we got plate 1, plate 2, and so on and so forth. Hit submit. And there we go. We're successful. And just with a click, here we are. So here is my plate set. We've got 4 plates, all of which are kind of very similar, with the goal being to generate a bunch of kind of replicate data for these 5 samples. And as I can see, just by kind of highlighting my samples, I can see that they've been placed into these wells. We've got some kind of concentration values here, with the goal to be performing a dilution curve for these plates. But other than this, other than just selecting the template, I didn't have to manually set anything else up. Using the template, Biologics LIMS actually does the bulk of the work for you. So reading that template can identify what wells these samples are going into, what wells are marked as control, what wells are marked as containing samples, and then kind of replicating that across the 4 plates. So it allows you to kind of have that idea of creating these repeatable templates that you can share with other team members to cut down on manual error. And from here, I can actually generate work list or instrument instructions that I could then take to my plate reader, and just kind of feed in and have it spit out kind of the assay data or the file that I'm going to use to import to track my assay data for this plate set. And so let's go ahead and do that. Let's import some assay data. We'll choose the relevant design. And here I am just going to drag and drop a file. It's a pretty generic kind of plate readout file that I've got. Just hit import, and here we are. Here is our data. So as you can see, we've got our sample IDs here, our well locations, so the wells that the samples are in, our plate sets, and then the individual plates that this data is associated with. And then, of course, we've got kind of our experimental values as well, alongside some calculated values of mean and standard deviation that we've done or that Biologics LIMS has done across these replicate groups. And then from here, you can kind of easily create visualizations, charts for further analysis. So here we've got kind of a standard curve fit for this data. And then Biologics LIMS kind of generates this statistical summary underneath. So tracking things like R squared, inflection point, slope, things like that. And then from here, you can actually kind of easily export this data for use in other tools or applications. So if you want to perform further analysis in any kind of external software, or if you want to stick this into a PowerPoint, or just easily share it with external collaborators, it's as easy as kind of exporting this chart as a PDF or PNG. And you can even take this information and place it directly into an ELN that you've been using to kind of track this entire process. So all the way from sample selection down to your results and interpretations. You can just stick it directly in. And because all of that data is linked, you could also include kind of references to the actual data as it appears in Biologics LIMS. And then another benefit of that data actually being linked and all of that data being related to one another is that it allows you to roll up your data. And so roll up your data from the sample level all the way up to any biological entities that are related to those samples. So what do I mean by that? Well, let's kind of quickly jump back to where this all started. So let's jump back to our Herceptin Fab molecule. So here we go. And you can see there's also this Assays tab. So if I click here, this allows me to see all of the assay data, so all of the experimental results for all of the samples that are associated with this molecule. So here are the kind of results we just imported. So that CBPA assay that we were doing. But here I've also got some additional assays, some protein yield quantifications, STS page assays for different samples that are associated with this molecule. So just with kind of one click, something as simple as navigating to a different tab, you can get this holistic idea of, okay, how is all of my data connected? And it allows you to ask questions of your data without kind of need of a data science team or the need to actually jump into your software and write SQL queries yourself. And yeah, you can easily see, from a given molecule, what samples do I have? What are their properties? And if you need to find a given sample to kind of obtain more material to use in kind of further analysis, you can easily do that. You can just click on the sample, and that information's right here. You can see exactly where it's stored. You can see the notebooks that it's been referenced in. And yeah, you can go right here to this kind of exact freezer location, grab more of that sample, and then perform further analysis. So this is just kind of a small glimpse behind the curtain. There's a lot more you can do with the product, even just with kind of plate-based assays. You can exclude outliers. You can more kits for kind of more iterative plate mechanics. Um, we barely kind of touched on storage. There's more you can do with ELN. There's a lot more here. But like I said, this was just kind of a glimpse of what's possible. But even hopefully, just with that glimpse, you could see the benefits of having an all-in-one solution, just like Biologics LIMS. And just as a reminder, those benefits are increased accuracy and standardization. So think kind of being able to track all of your data all in one place with full traceability and transparency. It's designed for ease of use and scalability as your lab grows. So it's easy to migrate, it's easy to learn, and it's easy to continue using kind of even as your lab and kind of needs grow and shift over time. And it empowers it empowers data-driven decision making. So whether that's through visualizations, searches, or roll-ups, you can quickly find the right data at the right time to answer your most important questions. So, what's next? Well, if you're interested in seeing a more targeted kind of demonstration of what biologics limbs can do, that's more tailored to your kind of specific science, just let us know. Reach out to us. We're more than happy to kind of work with you all through kind of what we have set up as our guided evaluation process through these three steps. So discover, kind of configure, and then trial. So this is kind of where our support that we offer organizations kind of starts. And yeah, we want to make sure that as you're going through your evaluation process, we're doing everything we can do to help and make sure you're successful and you get kind of that whole picture of how this can work for you. So you should be receiving a link via email to kind of schedule some time with us. So please don't hesitate to reach out. You can ask us any questions you may have. You can set up some time with us. And yeah, we can work together to kind of build a demo around your actual kind of samples, biologics entities, and your actual data. So with that being said, thank you all so very much again for joining. It was a pleasure kind of speaking with you all. And we hope to talk to you soon.